Hello everyone, this is BOC Science's Isotope presenting. Today we will talk about the stable isotope tracer technique. In 1912, when Joseph John Thomson made a determination with neon gas, no matter how the neon was purified, two parabolic traces were obtained on the screen of the mass spectrometer. Later on, Francis William Astron improved Thomson's mass spectrometer and further proved that neon indeed has two isotopes of different atomic masses, i.e., neon 20 and neon 22. In 1929, Stefan Meyring-Naud discovered nitrogen-15 in the absorption spectra of nitric oxide. In the same year, William Francis Gioke and Herrick Lee Johnston, while studying the absorption spectra of oxygen molecules, found that oxygen contains isotopes of atomic weight 17 and 18. In 1931, Harold Clayton Urey and others slowly evaporated liquid hydrogen at 14 K for residue spectroscopic analysis and discovered the hydrogen isotope with an atomic weight of 2 and named deuterium. The artificial production of nitrogen-15 was first reported by Urey and others later in 1937. In 1940, the biologically significant nitrogen-15, oxygen-18 and deuterium were produced in large quantities. In 1947, a symposium held at the University of Wisconsin regarding the theme, Application of Isotopes in Biology and Medicine, began a new era for the stable isotope tracer technique. In recent decades, the stable isotope tracer technique has developed rapidly. Carbon-13, deuterium, oxygen-18 and nitrogen-15 are widely used in biology, medicine, environmental protection, agriculture, microbiology, and other research fields. Several basic concepts are involved in the study of the isotope tracer technique. To elaborate, a nuclide is an atom with specific nuclear characteristics and refers broadly to atomic forms with different atomic numbers, mass numbers, and nuclear energy states. Isotopes are different nuclides of the same element with the same number of protons but different neutron numbers. Stable isotopes are isotopes with a stable atomic structure that do not spontaneously emit rays that would change the structure of the nucleus. Isotopic abundance is the relative percentage of stable isotope atoms of the element. Natural abundance is the isotopic abundance of an element in a natural substance. Atomic percent excess, also known as enrichment, is the difference between the abundance of an isotope in a specimen or marker and the natural abundance of that element. The isotope tracer technique is a research method that uses isotopically labeled nuclides or isotopically labeled compounds to track and study the movements and laws of related objects. Both radioisotopes and stable isotopes can be used as tracers. Radioisotopes, or isotopes whose nuclei are unstable and will decay, emitting alpha, beta or gamma rays. Common radioisotopes include hydrogen-3, carbon-14, phosphorus-32, sulfur-35, calcium-45, chromium-51, iron-59, iodine-125, iodine-131, silver-198, etc. Stable isotopes, that is, isotopes with stable nucleus structure that will not decay. Common stable isotopes are carbon-13, deuterium, oxygen-18, nitrogen-15, etc. The radioactive isotopes, or stable isotopes, and their compounds utilized in isotope tracing are chemically and biologically identical to the corresponding common elements and their compounds present in nature but only contain different nuclear physical properties. Therefore, it is possible to use isotopes as a marker for compound labeling, example, labeled foods, drugs, and metabolic substances. Taking advantage of the nuclear physical properties of radioactive isotopes, which constantly emit characteristic rays, it becomes sufficient to track their locations, quantities, and transformations within or outside the body at any time using nuclear detectors. Although stable isotopes do not emit rays, they can be determined by mass analysis instruments such as mass spectrometers, gas phase chromatography, and nuclear magnetic resonance by comparing the difference in mass with the corresponding common isotopes. The stable isotope tracer technique is used in various tracer studies, which relies on three basic principles. The first one relates to the isotopic composition of an element in nature is relatively constant. The second is that the isotope of one element is chemically identical and has tracer properties. The third refers to the mass differences between the isotope and that same element, and such differences in physical properties are detectable. The two stable isotopes of hydrogen are hydrogen-1, also known as protium, and hydrogen-2, also known as deuterium. Protium is the major stable isotope of hydrogen with a relative atomic mass of 1, which is mainly found in water and various hydrocarbons and has a natural abundance of 99.985%. Deuterium is another stable isotope of hydrogen, with a relative atomic mass of 2 and a natural abundance of 0.015%. The two stable isotopes of carbon are carbon-12 and carbon-13, with carbon-12 being the major stable isotope that contains a relative atomic mass of 12 and a natural abundance of 98.892%. Carbon-13 is another stable form of carbon isotope with a relative atomic mass of 13 and its natural abundance of 1.108%. The two stable isotopes of nitrogen are nitrogen-14 and nitrogen-15. Nitrogen-14 is the major stable isotope of nitrogen with a relative atomic mass of 14 and its natural abundance of 99.635%. Meanwhile, nitrogen-15 is another stable form of nitrogen isotope with a relative atomic mass of 15 and its natural abundance of 0.365%. There are three stable isotopes for elemental oxygen, which are oxygen-16, oxygen-17, and oxygen-18. Oxygen-16 is the major stable isotope of oxygen with a relative atomic mass of 16 and a natural abundance of 99.759%. Oxygen-17 is a stable form of oxygen isotope with a relative atomic mass of 17 and a natural abundance of 0.037%. Oxygen-18 is also a stable form of oxygen isotope with a relative atomic mass of 18 and a natural abundance of 0.204%. In comparison with the radioisotope tracer technique, the stable isotope tracer technique was advanced by the followings. First, the stable isotope tracer technique is not radioactive and has no radiation hazard, making it suitable for in vivo tracing, especially for pregnant women and children. Thus, no special protective measures are necessary for its use. Second, it is safe and reliable because stable isotopes, such as carbon-13, nitrogen-15, oxygen-18, etc., are generally not chemically toxic. Third, it does not pollute the environment and its experimental range is not restricted by environmental conditions. Fourth, stable isotope-labeled compounds will not undergo radiation self-decomposition nor decay, thus not limited by time and suitable for long-time tracing. Last but not least is that the stable isotope tracer technique can carry out experiments that are difficult to be performed when using the radioisotope tracer technique. For example, nuclides such as nitrogen and oxygen do not include radioisotopes of short half-lives. Stable isotopes are the only available tracers. When stable isotope tracer analysis is performed using mass spectrometry and nuclear magnetic resonance spectrometry, molecular structures of tracers can be observed via the labeling positions. In contrast, in terms of radioisotope analysis, complex chemical degradation and the separation of degradation products are required to determine the molecular structures. Therefore, stable isotopes can be dual-labeled with radioisotopes to compensate for the lack of radioisotope labeling alone. Technically, the stable isotope tracer technique was known to use in two classical applications in biology. In 1957, Meselson and Stahl cultured E. coli bacteria in mediums containing nitrogen-15 only for growth and replication. After generations, when the DNA materials of these bacteria contained only nitrogen-15, mediums were replaced with the nitrogen-14 only medium for another generation. Then samples were taken at different times, and DNA was extracted for density gradient centrifugation. As a result, it was found that the molecular density of the extracted DNA samples decreased from generation 0, heavy density, to generation 1, medium density, located between nitrogen-15 and nitrogen-14. That said, there are equal densities of nitrogen-15 and nitrogen-14 contained by DNAs. If replication is fully conserved, then only two types of DNAs, nitrogen-15 and nitrogen-14, will be present. Thus, these experimental results confirm the semi-conservative nature of DNA replication at the molecular level. Meanwhile, the second application involved the confirmation of oxygen sources in photosynthesis. In 1939, Rubin and Kamen labeled water and carbon dioxide separately with stable isotope oxygen-18. They then performed two sets of comparative experiments. One was provided with water and oxygen-18 labeled carbon dioxide, and the other was supplied with oxygen-18 labeled water and carbon dioxide. All other conditions remained the same. In the end, it was analyzed that the first experimental objective released oxygen without isotope labeling, while the second experimental objective released oxygen with isotope labeling, thus providing strong evidence that oxygen released by plant photosynthesis derived from water rather than carbon dioxide. BOC Sciences is an innovator and manufacturer of isotope-labeled materials, and we are committed to becoming the leader in the isotope industry. For more information about BOC Sciences isotope products and services, please feel free to contact us.